Parkinson's disease is a neurological disorder characterized by the progressive loss of dopamine-producing neurons in the brain. The decreased level of this chemical messenger affects the activity of the central nervous system and, in turn, the body's movements. It leads to a range of symptoms, including slow movements, tremors in the hands and other body parts, stiffness in the limbs, changes in posture, and difficulties with balance. While medications like dopamine precursors, which convert to dopamine in the brain, are prescribed to alleviate the symptoms, a complete cure for Parkinson's disease still does not exist. As one of the most prevalent neurodegenerative disorders, Parkinson's impacts a significant number of individuals in India. Estimates suggest around 10 to 15 lakh affected individuals presently exist in India. This number will increase many folds in coming decades. One of the underlying causes of Parkinson's disease lies in the misfolding and accumulation of a protein called alpha-synuclein. These misfolded proteins clump together to form aggregates known as Lewy bodies, which are found within the neurons of Parkinson's patients. When examined under a microscope, Lewy bodies appear spherical clumps. It is now known that the misfolding and aggregation process leading to the formation of amyloids and neurodegeneration precedes the manifestation of motor symptoms by several years. Some of these amyloids, also referred to as seeds, manage to escape the protective blood-brain barrier and enter the bloodstream. However, their concentration is too low for accurate detection by conventional methods. The recent advancement of neurological disorders detection from blood and CSF is the development of seed amplification assay, where ultra-low levels of misfolded proteins are amplified to aid the detection. This is similar to the prion replication principle for infectious prion disease and is used for its detection. The principle of seed amplification assay can be applied to detecting alpha-synuclein in Parkinson's patients. However, the challenge arises because of the unstructured nature of the wild-type alpha-synuclein protein, which itself tends to convert into amyloid-like structures in this assay, potentially leading to false positive results. To mitigate this issue, we have developed a blood test that utilizes a modified substrate. This substrate is created by engineering an alpha-synuclein variant that lacks self-aggregation propensity but readily aggregates in the presence of the misfolded protein seeds. To carry out this blood test, we collect blood samples from individuals with Parkinson's disease and separate the amyloid-containing serum by centrifugation. Then, we add serum to the solution of modified engineered protein, which acts as a substrate. Then, this mixture undergoes multiple rounds of incubation and sonication, similar to the PCR cycle. Sonication breaks down the aggregates into smaller fragments that exhibit further seeding to amplify the amyloids. After every defined cycle, an aliquot is taken and centrifuged to separate the alpha-synuclein aggregates, which can be analyzed using Western blot, thioflavin T assay, which shows increased fluorescence and quantifies the amyloid formation, and by the Congo red dot blot assay. In Congo red dot blot assay, before washing with ethanol, all the spots show a red signal. However, after washing, only the patient serum with amplified seed concentration will retain Congo red, and the healthy control sample shows no signal. Importantly, this technology represents a significant advancement in detecting Parkinson's disease, which currently lacks precise diagnostic methods. With this approach, easy-to-administer and cost-effective tests can be conducted with a more than 90% accuracy rate. This breakthrough holds immense potential for improving patient care and management of Parkinson's disease, offering hope for earlier detection and intervention.